Smooth muscle. If skeletal muscles are found attached to our bones and sometimes skin, smooth muscles are always located in the internal organs, usually hollow organs, those that have space inside, except the heart. Example is stomach. Stomach has 3 layers of smooth muscles, and when they contract or relax, they mix food with the stomach juice. Next one, intestines always have smooth muscles in their walls that allow them to move the food along them and allow absorption. Some other organs that include smooth muscles would be urinary bladder, gallbladder, bile ducts, bronchi in our lungs, and also blood vessels. In the walls of blood vessels, there are smooth muscles. When they contract, they maintain blood pressure. So smooth muscle cells are single cells, not striated, and each of them has only one nucleus. That's how they look under the microscope. No striations. When small muscles of some hollow organ, usually tube-like organ, contract and relax in turn, they provide peristalsis, movement of something, usually it's food, bolus of food, through esophagus or small intestine. And for that, there are 2 layers, longitudinal layer of muscles and circular layer. So they contract and relax and turn in turn, and they move that bolus of food through the intestine or through the esophagus to the stomach. Microscopic structure. Smooth muscles are spindle-shaped, one nucleus, and it's much thinner and shorter compared with skeletal muscles. There is only one layer of connective tissue covering that muscle. It's called endomysium. There is some sarcoplasmic reticulum, but much less than in skeletal muscle. Instead, there are special structures right here, cavilli. They work as a calcium storage, and they allow smooth muscle to get calcium from the fluid outside of the muscle cell. There are no sarcomeres, no myofibrils or T-tubules, so the whole structure is simple. There are still myofilaments in smooth muscles, thick myosin and thin actin filaments, but the ratio of them is different, 1 thick filament to 13 thin filaments, while in skeletal muscle that ratio is 1 to 2. There is no troponin or tropomyosin proteins, remember those were attached to actin in skeletal muscles. Instead, calmodulin protein binds calcium for contraction, for forming bridges between actin and myosin. Those actin and myosin, myofilaments, are arranged in a spiral. So when smooth muscle contracts, it contracts like a corkscrew, a little bit twisted. There are dense bodies on the surface of a smooth muscle cell. Those are proteins that anchor intermediate filaments to their cell membrane, sarcolemma. And inside, if we look deeper, those are dense bodies, proteins. Thick filaments or myosin are attached, thin filaments or actin are attached to them, and between them there are thick filaments. So when thick filaments using their heads start pulling thin filaments to each other, this part shortens, but it's not a sarcomere. Single units are motor units of smooth muscles. What does it mean? There is one neuron that branches, and that is not a motor neuron, not like in skeletal muscle. Skeletal muscles have motor neurons, and we can voluntarily control our skeletal muscles. Raise our hand, for example. But we cannot do to smooth muscles because right here there are autonomic nerve fibers. We cannot control them. Those autonomic nerve fibers branch, and each branch has several bulges called varicosities, and those vesicles contain neurotransmitters, and each of those varicosities activates one smooth muscle cell. Contraction of smooth muscle is slow comparing with skeletal muscles and very energy efficient. Sometimes myofilaments, actin and myosin in smooth muscle can even contract and stay in that position for a long period of time without using ATP. That is called latch state. That's how muscles maintain muscle tone that allows, for example, our blood vessels to be partially contracted all the time to maintain our blood pressure.